All right, so chapter 14 starts our biochemistry section of all of this, and we're going to start by talking about carbohydrates. So carbohydrates are also called sugars or starches, and these are polyhydroxyaldehydes or ketones. So you can, right off the bat, see why we wait and do the biochemistry after we talk about the organic chemistry, because we're going to build on some of these organic fundamentals that we learned, particularly with these different functional groups. So polyhydroxy means that you're going to have multiple OHs, multiple OH groups, and then aldehydes or ketones. If you remember, aldehydes are when you have a C double bonded to O bonded to an H, and a ketone is when you have C double bonded to O with carbons on either side. And you would have another carbon attached over there in your aldehyde. So it's going to be important to make sure we understand these functional groups back from our organic chemistry back in chapter 10. Within the carbohydrates, we can have monosaccharides, disaccharides, polysaccharides that basically describe how many sugars are attached to each other, one, two, or many. Carbohydrates are actually synthesized in photosynthesis. So within photosynthesis, plants, they make mainly 6-carbon sugars, but in the process of making 6-carbon sugars, they actually make shorter sugars as well. But the whole point of plants taking in sun and carbon dioxide, so plants take in CO2 and they use energy from the sun to make water, as well as glucose, which is one of the sugars, as well as oxygen. Physiologically, for us humans, we use these carbohydrates for bursts of energy that we need during exercise and for a lot of other things we do throughout the day. So again, those bursts of energy comes in the form of glucose, which we then use that glucose to metabolize it down into ATP, which is our cellular source of energy. So this just goes over the whole process of photosynthesis. So you can see that balanced chemical equation. You have 6 CO2s coming in to give you the 6 carbons that are present in glucose. So carbohydrates are often thought of as carbon with waters attached. And however many carbons you have, you're going to have the same number of waters. So you can see that for glucose with 6 carbons, we would have like 6 water molecules. So C6H12O6, you're always going to have the same number of carbons and oxygens, and you're going to have twice as many hydrogens. And as we look at the structures of these sugars coming up soon, you'll be able to see this a little bit more clearly. All right, so monosaccharides, these are the simplest carbohydrates, and they're generally going to have anywhere between 3 to 6 carbons. Those 3 to 6 carbons are going to be linked together with a carbonyl group somewhere in there. If your carbonyl is at C1, so if it's at that first carbon, that's going to be an aldose, because if you have a carbonyl at C1, that's going to be an aldehyde. And then if it's an aldehyde and a sugar, we identify that as an aldose. So it's like a combination of an aldehyde, and then anytime that something ends in ose, that's the suffix for a sugar, so an aldose. If your carbonyl is at C2, then we have what we refer to as a ketose, because then you have like a ketone, a ketone sugar. So a ketose would be a ketone sugar. All right, so here's a couple examples of these. So the simplest aldose is going to be a 3-carbon sugar, and that's called glyceraldehyde. This CHO group that you see kind of up here, right, that's going to be an aldehyde, right? So another way you could draw that would be C double bonded to O, bonded to H, bonded to the COH, H, and then down here would be C. And instead of drawing out the H2OH, I'll write it out this way. That's CH2OH. And then for the dihydroxyacetone, that's going to be your simplest ketose. So it's a 3-carbon sugar that's a ketone, and you can see the ketone part there where you have the carbonyl group with carbons on either side. So these are both have the formula of C3H6O3, and because they have the same formula but they're connected differently, we refer to those as constitutional isomers, right? Which you learned back as a definition within in your organic chemistry chapters, chapter 10 specifically. A monosaccharide can be further characterized by the number of carbons it has in its chain. So a triose is going to have 3 carbons, a tetrose 4, pentose 5, a hexose 6. So if we were to go back and look at glyceraldehyde, right, we could say glyceraldehyde is an aldotriose because it would be an aldehyde, right? So that's the aldo part, and then triose would mean it has 3 carbons. Whereas dihydroxyacetone would be a ketotriose. Keto because it's a ketose, and then a triose because it has 3 carbons. For instance, glucose, like the one that the plants make, that would be an aldohexose because it would be an aldose that's 6 carbon long. So it would be an aldohexose. Monosaccharides in general are sweet, right? That's the whole idea of a sugar. Sugars taste sweet. Their relative sweetness varies a great deal, so depending on the specific monosaccharide you're looking at, the actual taste and the relative sweetness is going to vary. These are going to be polar compounds because of the multiple OHs that they have. So the polar compounds primarily because of all the OHs that are there. So because of that, they're going to be soluble in water because those OHs can hydrogen bond, right? And they're going to be water soluble and have very high melting points because of those strong interactions that can form. Okay, so with monosaccharides, we need to identify a new term. We're going to identify this term chirality centers. So a chirality center refers to a carbon that has 4 bonds. So a lot of times carbons are going to be chirality centers. And the idea of a chiral center means you have a carbon attached to 4 different things. So if you're this carbon in the middle where the arrow is, you can see if it looks to the right, it sees an OH. Right? And if it looks to the left, it sees an H. And if it looks up, it sees a CO or CHO, right? It sees more than just a carbon. So it sees that whole group. It sees a CHO or C double bonded to O bonded to H. It sees the aldehyde part, if you will. And then on the bottom, if it looks down there, it sees a CH2OH. So the carbon in the middle is a chiral center because it has 4 different things attached to it. So all 4 ways it looks, it's going to be different. So anytime something has a chiral center like that, we can further look at that carbon in terms of its stereochemistry. In other words, where those particular groups are in relation to each other matters. So with glyceraldehyde, which is what we're looking at here, we can say it has 1 chiral center, and it has 2 possible enantiomers. So this word enantiomer refers to a non-superimposable mirror image of each other. So the idea here is that D-glyceraldehyde, which is the one on the left, is different than L-glyceraldehyde, which is the one on the right. So if you were to look at it, the mirror image, right, of the one on the left has the OH on the right, or on the left, and the H on the right. So this is considered an isomer, or specifically an enantiomer, because it's a non-superimposable mirror image of each other. And the idea here is that no matter how, like if you were to look at the tetrahedral shape of this, you can't make L-glyceraldehyde. You can't just twist it around or whatever and turn it into D-glyceraldehyde because of the location of that H and the OH are going to be different in relation to where those other 2 groups are. So D-glyceraldehyde is different than L-glyceraldehyde, and it depends on the relation of those groups in terms of where they are to each other. So generally speaking, what we do for this is we look at we take the carbons and we put them in a line, which is what I'm circling over here in the molecule on the left. So those carbons all go in a line. For D sugars, the OH is on the right. For L sugars, the OH is on the left. So that's how we distinguish between D and L. So for D-glyceraldehyde, the OH is on the right. For L-glyceraldehyde, the OH is on the left. Okay. The other thing to remind you guys from this is the difference but what these wedged and dashed lines mean. Remember, the wedges mean that they're coming out towards you. The dashed lines mean they're going away from you. With sugars, we typically, or not typically, but frequently draw things in what are called the Fischer projections, which is what's shown over here on the right. I like to describe these Fischer projections as like a modified type of skeletal structure, with the idea being that the region where you don't see anything and it's just a bunch of lines intersecting right there in the middle, right? Just like in a skeletal structure where lines come together, there would be a carbon there. The idea with these Fischer projections in particular is that anywhere where you have those lines come together like that, you're going to be looking at a chiral carbon. So it's going to be specifically a carbon that is a chiral center, right? Whereas this carbon and this carbon are not chiral, right? The one on the bottom is not chiral because it's attached to, if I were to draw it out, it has CH2OH and then attached to more. Because that H and that H, because those are both H's, it's not chiral. To be chiral, it has to be attached to 4 different things. Because those 2 H's are the same, it's not chiral. And then if we were to continue drawing this, the reason the one up top is not chiral is because it has a double bond, right? So that carbon up top here is also not chiral because it only has 3 bonds. To be chiral, you have to have 4 bonds. Okay, so what happens if you look at something like glucose, for instance? So something like glucose is going to have 4 chiral centers. So if you look at the Fischer projection over on the right, you can see that anywhere where you have those carbons that have the asterisks, which are chiral centers because they're all attached to 4 different things, you have 4 different chiral centers. So how do you know if this is D-glucose or L-glucose? Because we said if the OH is on the left, it's L. If the OH is on the right, it's D. But in this case, we have 4 different OHs. So in this one, the way that we determine and distinguish D versus L, what you do is you find the carbonyl carbon. So the carbonyl carbon is going to be your C that's double bonded to O, which is going to be this one up here. So that's going to be your carbonyl carbon. So your carbonyl carbon is going to be the one that is C double bonded to O. All right? So your carbonyl carbon, you find that, and then you find the chiral carbon farthest away from it. So in this case, you go the farthest away chiral carbon, and you come down there. And you say, on that particular carbon, is the OH on the right or the left? In this case, it's on the right. So you would say this one is D-glucose, and the other chiral carbons don't matter. So you find the chiral carbon farthest away from your carbonyl carbon, and that's how you determine whether it's a D or an L sugar. All right, and that's the same thing that this slide here describes. So again, OH on the right is going to be D, OH on the left is going to be L. Now, naturally occurring sugars, so any sugar that's occurred in nature, whether it's through like a plant synthesis or the sugars that we make physiologically, the OH is always going to be on that right from the carbonyl carbon. So naturally occurring sugars are always going to be D sugars. So L-glucose exists, it's just not a naturally occurring one. It can be synthesized in a lab, but it's not something that our body makes naturally. So a couple common monosaccharides, or really one common monosaccharide that is most prevalent is going to be glucose, which is often also called dextrose. This is blood sugar. So if you go to a lab to have blood work done, they'll measure your blood sugar levels, and sometimes the reading will give you a blood glucose level, sometimes it'll be a blood dextrose level. It's the exact same thing. Typically those values are in the 70 to 110 milligram per deciliter range. More recently, in like blood work I've had done, for instance, it says 70 to 100 milligram per deciliter. Once you start to get above 100 milligram per deciliter, they start to worry about diabetes. So speaking of diabetes, that's often controlled by, or largely controlled by, insulin. So insulin is a hormone that helps regulate your blood sugar levels. And what insulin does is it stimulates your cells to take glucose into tissues and convert it to something called glycogen. So glycogen is basically a whole bunch of glucoses linked together, and we're going to talk about that later in this chapter as a type of polysaccharide, but that's basically our way to store glucose for future use. So patients that have diabetes don't produce insulin adequately, so they have trouble regulating their blood sugar levels, so they have to monitor their diet and typically inject insulin themselves to help monitor that. Another common monosaccharide is going to be galactose. So galactose is one of the components of lactose, which is a disaccharide. And we'll talk about disaccharides in the next video. But disaccharides have two monosaccharides linked together. In lactose, you have a galactose and a glucose linked together. So galactose is a common monosaccharide, and there are some people that have something called galactosemia. And people who have galactosemia have they lack an enzyme that is necessary to metabolize galactose, which, if that's the case, the galactose is going to accumulate, and this can cause cataracts and liver cirrhosis, which can be very problematic. Another monosaccharide that is really common is fructose. So fructose, I want you to know, is a ketohexose. So fructose is by far probably the most common ketose that we'll talk about. And it's a component within sucrose. So sucrose is a disaccharide that is a mixture of fructose and glucose. But fructose is very sweet. It's a ketohexose that's found in honey and is really sweet compared to, for instance, like table sugar. All right, so just taking a look at this structure here, if I were to ask you, is this a D sugar or an L sugar? How would you identify it? Well, first thing you do is you find the carbonyl carbon, which is going to be right there. And then you want to find your chiral carbons. And you would say that's a chiral carbon, that's a chiral carbon, and that's a chiral carbon, right? The CH2OHs on the top and the bottom are not chiral because they both have carbons that have two hydrogens attached to them, whereas all these others have an H on one side, an OH on the other side, and then different carbon groups on the top and bottom. So to find if it's D or L, you find the carbonyl carbon. You go to the chiral carbon farthest away, which is going to be this one down here. The OH is on the right, so that means it's going to be a D sugar. If it's a D sugar, that also means it's going to be naturally occurring, right? So those two we can answer both at once. To determine whether this is an aldose or a ketose, we would define this one as a ketose because where we have that arrow is pointing at the carbonyl, and if we look at that functional group, we would say it's a ketose. All right, so we could also call it ketohexose because it is a 6-carbon long sugar. And specifically, this ketohexose would be fructose.